Welcome to the course on basic genetics. In this course, there are three teachers, Dr. Richa Arya, Professor Madhu Gavaldas Tapadia, and myself Jagat Kumar Roy. I am retired, and the other two, they are from Department of Zoology, Banaras Hindu University. This is week 12, lecture number 4, regulation of gene activity in eukaryotes. This subtopic is RNA processing. We have already seen that how transcription occurs, how RNA is formed. This is once again to show you the same thing, that this is the DNA that is opened, this is the DNA that is opened, and this is the RNA polymerase that is synthesizing the RNA, and this is the new RNA that is formed. So, this is 5' side of the nascent RNA, and this is 3' side, which is the site where the new bases are being added. I did say in my last lecture that this sequence, the sequence on RNA and the sequence in the DNA in this strand, they are same, and hence this strand is called as plus strand, this strand is known as sense strand. Now, when RNA is made, once RNA is made, that is when the RNA is being formed, its 5' end is free, this 5' end is modified. This process is called 5' capping. Essentially, this is to seal the 5' end so that this is no more available for any enzymatic activity. When I was showing the picture, I was showing that this is the base that is being added, I was showing. Now, here what has happened? To this base, a G is added, that is a guanine nucleotide triphosphate is added in the reverse direction. That means this is 5' end of the first base, and this is 5' end in this direction is the guanine nucleotide that is being added. So, this is the guanine nucleotide, this is the sugar, and this was the first base, and this is the sugar of this. The 3 phosphates, 2 phosphate, this had initially the 3 phosphate, and this also initially had the 3 phosphate. So, 6 phosphate, out of which now 3 are remaining, 3 have gone in the process. Okay. So, this is guanosine nucleotide addition. Then subsequently what happens, that there is methylation. So, this is methylated. Once this is methylated, methylation is associated with the inactivity, with the tight or with the condensed condition. So, once this is methylated, this side is better protected. Because this is methylated in all, you call it as cap 0. Then this one is now substrate for next methylation. So, where is the next methylation? The next methylation is in the sugar of the first nucleotide. So, here OH was there because this is RNA. So, this sugar had OH here, 2' OH, and here methylated group, that is CH3 group, is added. So, this is called as cap 1, this is M7 GPP. Then after that, so these two methylations, this is common in all, this is also common in most. A third methylation, that is here in the second base, if the second base is A, first base is usually A. This base is usually A, that is adenine. And if the second base is A, then there is another methyl group that is added, and this is known as cap2. So, with this, now this is absolutely protected from the 5 prime site. Then, when the RNA is formed, so far what I was showing this condition, that when the transcription was going on, I was showing that situation. In last class, I did talk that when the RNA is formed, the 3 prime end is generated. That is, the synthesis continues. At one place, this is cut, and there is poly-A polymerase that adds poly-A's, that is several A's, approximately 200 A's are added. So, on the 3 prime end, you have poly-A or AAAAAA. Some of the RNAs are not polyadenylated, so they have slightly different kind of protection, but otherwise most of the mRNAs, they are poly-A plus. When poly-A is there, that is 200 base of A is added, you call it as the poly-A plus mRNA or poly-A plus hnRNA. At this moment, I will call it as heterogeneous nuclear RNA, because this RNA is not in its final condition. This will get further modified to become the final messenger RNA. So, the next thing occurs, that is splicing. Where the splicing occurs? The splicing occurs when the RNA is formed. When it is inside the nucleus, then splicing occurs. What is splicing? That any RNA has exons and introns. Exons means a length of nucleotide, they will be retained in final mRNA. Introns are the intervening sequences, that means those sequences will go out. That means if it has exon 1, then intron 1, then exon 2, then intron 2, exon 3, then intron 3. Exon 1, exon 2 and exon 3, they will come together and introns will be looped out and spliced. The question is now how splicing occurs. This is a wonderful mechanism that these things, the intervening sequences removed. There are 3 different types of splicing, or the splicing event has been grouped into 3. One, you call it as group 1 splicing. The example given here is rRNA gene of protozoan Tetrahymena thermophila. So, this rRNA undergoes splicing, and here how splicing occurs? You can see this is the nucleotide, that means this is the RNA that is formed. Now, at the point where splicing has to occur, a G guanine nucleotide that comes. It can be a guanine nucleoside, it can be a guanine nucleotide. So, this comes. If it comes, this is O. O has partial negative charge. So, this has partial negative charge and P has partial positive charge. So, that means there will be an interaction with O and P. When there is interaction, what will happen? That this is bonded here and this becomes free. So, this becomes OH and this becomes free. So, this is out now. So this is out, and G, this has got now inserted with this. So this now becomes the first nucleotide. So this is part of the exon, that this is part of that region which has to be staying. So this is exon. This is exon 1. And this is intron. Now because this has OH, this you can see this OH. Wherever the intron ends, if this touches there, this can again form bond, and this part, so this will be OH and this part can be out. This part can go out. So this way the introns are removed. Once again you can see here that this is exon, this is intron, this is exon. Here a G that is attacking this, and the result is G is part of this now. So G is now part of this, and this is free. So this is free, this part. This is the exon 1. And now this exon at the end of intron, again it makes a nucleophilic attack. So now this and this, they came together, and GOH, this is removed. From there G can be removed and this can become a circular one. So this is group 1 splicing. Now with this, let us go to another kind of splicing, that is group 2 splicing. Where group 2 splicing is found? This is found in bacteria. It is not that all the bacterial genes are spliced. It is not that all mitochondrial genes or RNAs are spliced. It is not that all chloroplast RNAs are spliced, but some of them do get spliced. Wherever the splicing is there, the group 2 splicing mechanism works. If I show here this picture, the first part, this is again same as we were seeing in the earlier picture. Just to emphasize that this is 2 prime. This is 1 prime carbon, this is 2 prime carbon, this is 3 prime carbon, this is 4 prime carbon, this is 5 prime carbon. So this 2 prime carbon, this is 2 prime. This 2 prime carbon, this has OH in RNA. In DNA, this is deoxy, so this is having only H, both side HH, and here this is OH. As I said, that OH group, if it is there, this can have a nucleophilic attack with P. So that is what happens. So here I am showing you group 2 intron. This is first exon, this is intron, this is second exon. In the intron, now the question is, if this happens, that means all the OH which are present in bases, they can have nucleophilic attack? No, it is not so. It is the secondary and tertiary structure that is important. The tertiary structure is such that only 1 or 2, or mainly the 1 base OH is exposed. That is this part, this AOH, that is one of the A in the middle of the intron, whose sugar has 2 prime OH. So this is exposed out. And in the 3 dimension, where it is, this is exactly lying near the end of the exon. So then what happens? This makes a nucleophilic attack. When it makes a nucleophilic attack, this becomes separate and this becomes 3' OH. After the nucleophilic attack, this forms a lariat like this. Now, this free OH again makes an attack at the base of the intron or at the beginning of the exon. So, result is this, the two introns got joined and in between this is out. This is called as a lariat. This is called as lariat. Okay. So, we saw these two. One was in the tetrahymena, the other was in some of the bacteria and in some of the organelle genes, they have these splicing. Always, once again I will say that these splicings, there is nothing which is guiding, there is no guide. But here the tertiary structure is very, very important. And in this, whether I was talking about the group 1 splicing, the question is who will attack here? Which one? The OH means what I am asking, that this GOH, where this will attack? This will attack only at that place where in the 3-dimensional structure this is best exposed part. So, that means there is no ambiguity, this will attack at a given region that gets spliced or that gets sliced at that point. OH is now there. Again in the 3-dimensional structure, this OH which is now free, this OH means this is the part which is now free or this is the part which is now free. This will attack at a given place in this, I am talking about this OH. This will attack at the end of the intron or the beginning of the exon. I will say beginning of the exon because there is a P there, and thus there is nothing that it will occur at any place, this will occur at specific place only. The third one is the nuclear splicing. All the eukaryotic genes, they have splicing and that occurs in the nucleus. So, you can see here that this is the RNA which is being formed. So, this is the RNA being formed and this is the splicing that is taking place when the RNA is being formed. It is not that RNA has to form, come out, become mature, nothing like this. So, this is in the nucleus. In the nuclear splicing, if I give you this simple diagram that this is one exon, this is another exon and this is the intron. The intron at 5' region, this has a GU, and this intron at the 3' region, this has AG. So, you call it as the intron has a boundary here. GU, it starts with GU and it ends with AG. So, it is called as GUAG rule. And so, this you call it as donor, this G or this region, you call it as donor because this will allow this splicing to occur, this will donate its group here, and the other end which will get cut, you call it as acceptor. This is 3' splice site or acceptor. In between this, there is a that is there, and the 2' OH of this A, this will have the nucleophilic attack with this. So, the situation is that first this will get cut, this will form the lariat, this its end will attack here, and this will become continuous, and this lariat is separated. Once again, in last projection, I did say this is the lariat. So, this is the structure that is there, and that is GU, and this is AOH, this has attacked this. So, how precisely these things are happening? In nuclear splicing, there are several factors, they guide this. So, they make a structure called spliceosome. You can see here that this is the pre-mRNA, or this is called as hnRNA, heterogeneous nuclear RNA. This is exon, this is intron, and this is the middle portion. So, first there are certain RNPs, those are added. So, this is U1 that is added at the beginning of the intron, that is 5' splice site. U2 is added towards the end, that is 3' splice site. So, these two are added. These proteins, they have protein-protein interaction also. So, this becomes a pre-spliceosome, and then there are 3 together, U4, U5 and U6. So, these 3 snRNP means this is RNA with the protein, and they are present, and all of them, they are together because there is RNA and there is base pairing between them. So, U4, U6, they will have base pairing, U5 will also have some base pairing, and thus they are there. Once they come here, they are now mounted on this. When it is mounted, so you call it as complex B. Here you have all the U1, U2, U4, U5, U6, all are there. Then next what happens, that U1 is out, U4 is out. Once this binding occurs, always the bindings are such, when 2 are bound, maybe the interaction with the third is less and the third can go out. So, this is how these 2 are out. Now, you have U2, U5 and U6. So, this is the spliceosome that is there, and you call it as complex B plus. Then when this happens, then next step is further where the splicing will occur, that is cutting and joining. So, what these proteins are doing? These proteins are actually making them join together. So, I said that there is a GU. So, at that place there are proteins, and those proteins will fold it and will get touched with the A. So, now GU is in touch with A. This is the loop kind of a situation that happened. So, once that is there, then the splicing is occurring, a lariat is formed. When the lariat is formed, the other part, the end of this intron, they will have the attack, and result is the mRNA is formed. This is the mRNA that is formed, and this is the lariat that is out. This is the lariat that is out. And all the 3, they were present, all the 3 snRNPs. This is small nuclear RNA with protein. They are out and thus splicing occurred. So, these are the spliceosome complex and the splicing occurred. So, with this I will come to the activity and discussion question. What is the importance of 5' capping and 3' polyadenylation for a transcript? The answer to some extent I have given. This is for the protection of this RNA. Then the next thing, that is very important question, this you have to find out the answer, that why did splicing evolved? Why it was required that some of the nucleotides they are present, they have to be removed to finally form the thing. So, this and whether this was a design to keep the gene sequence secret or what, we have to find out this. So, with this I stop and I thank you very much.